Dear pediatrician colleagues and friends, thank you for inviting me to present this interesting topic. Thanks to PEGHAN. My name is Tushar and I work as a pediatrician with interest in pediatric allergy and gastroenterology in Pilgrim's Hospital, Boston, Lincolnshire. First of all, please accept my apology as I couldn't attend the study day in person and forwarded the recorded presentation. However, I will try my best to make some sense of the pathophysiology and underlying mechanism that define food allergy. I have no conflict of interests. This is a diverse research active topic and we are learning every day. The influencer of the immunopathology of food allergy is complex. To keep it simple, I will avoid complex details of receptor and chemokines that participate in the intracellular signal pathways and intercellular communications. Multiple immunoactive cells play their well-defined role in the business. As a clinician, we should understand the basic pathophysiology and try to link the pathophysiology of allergy with the clinical presentation as well as therapeutic aspect of current treatment. Aim of this presentation is to discuss the basic pathophysiology and immunophysiology, how immune system works, key players in allergic inflammation, mucosal and gut immune system, therapeutic and clinical implications of immunopathology. Please feel free to forward the questions in our PEGHAN WhatsApp group or message me directly in Twitter, and I will try my best to answer best of whatever little I know. In the end, I have included some journal article which you may find useful for further reading. I must emphasize the fact that when we talk about allergy, we are talking about allergic inflammation. Understandably, there is overlapping of mechanism when we talk about IgE and non-IgE mediated allergy. Let us refresh some of the basic terminology linked to atopic inflammation. As we all know, we are born with a degree of immunity to survive soon after birth in a hostile world full of pathogen. As the time goes by, a newborn matures into a toddler, a child, and subsequently an adult. The immune system adapts itself and learns how to deal with harmful bugs. This immunological learning is either by immunization or real life infection, which is called acquired immunity. So, what happens in allergy? Our immune system evolved over millions of years to kill harmful pathogen by identifying the foreign cells and foreign cellular fingerprints. In allergy, the same immune system misidentify a relatively harmless protein as harmful antigen and initiate an inappropriately severe response. The basic of any allergic reaction is IgE antibody mediated reaction, where mast cells, Th2 cells, and B cells are working together. This misplaced immunological response could be secondary to pro-inflammatory microenvironment within the gut, misunderstood intercellular signaling, and inability to control inflammation because of genetically determined faulty anti-inflammatory mechanism. Effect of dysfunctional bowel ecosystem made from pre, pro, and postbiotic. Therefore, it is not a straightforward business. There are several other situations where both IgE mediated and non-IgE mediated mechanisms act together, leading to interesting clinical presentation. One such example is EOE and F5, where several arms of immunological mechanisms are possibly working together and standard investigations for allergy is not helpful. To summarize, food allergen is a mistaken identity and cellular inflammation is directed against innocent bite. As I discussed in the last slide, successful removal of pathogen by early recognition of microbes by the components of the innate immune system saves us from severe infection. These involves the complement system, specialized receptors expressed on natural killer cells, and the family of toll-like receptors that are expressed on myeloid as well as lymphoid cells, which have the ability to recognize specific microbial molecular structure. Successful engagement of some of these pathways leads to an inflammatory response with destruction of the pathogen. The enlistment of BC, that is dendritic cells, and T cells and or B cells interactions are pivotal, and in later sections of presentation we will explain the role of these cells in allergic reaction. And well-orchestrated innate and adaptive immune response will lead to pathogen eradication and host immunity. This is how infection is dealt with. TLR or toll-like receptors are expressed in innate immune cells such as dendritic cells and macrophages, as well as non-immune cells such as fibroblast cells and epithelial cells. TLRs are largely classified into two subfamilies based on their localization. As I mentioned, all these cells dedicated to protect from microbial invasion is closely linked to pathogenesis of allergy. We will learn in the subsequent slides that there are several layers of defense in which is highly efficient and evolved immunological organization. The first layer is deployed rapidly to engage the invaders soon after antigen from bacteria or food allergen in case of an allergic reaction enters our body, whereas subsequent more complex reactive defense mechanisms take longer time to engage in action. Memory of previous engagement with similar pathogen stays in immunological memory, and subsequent invasion trigger more robust and quicker defense against the pathogenic invader once the initial responder are engaged and exhausted. In case of food allergen, the immune system deal with food-derived antigen in a similar manner. The only difference is the immune response is skewed towards humoral or Th2 skewed IgE response. Understanding the mast cell is a must for understanding allergy. However, this is an elusive cell which is difficult to study in vitro, and how it functions in vivo is based mainly on animal studies. This is a cell of myeloid lineage and hyperexcitable first-line defender, a key player linking both innate and adaptive immunity. They are found in abundance where environment continuously interacts with epithelial barrier. This essentially means that they are present in GI mucosa, under the skin, and the respiratory lining, where our body is bombarded with both pathogen and allergen. Once mast cells are activated at the site of inflammation, they recruit more mast cells, leading to release of significant amount of histamine. The mast cells are indestructible. That essentially means that after releasing histamine, they can regranulate without dying out, a unique feature amongst first-line defender. Mast cells are activated by antigen and IgE antibody complex, and tryptase is released alongside histamine. Both histamine and tryptase are packed together within the mast cell. Histamine induces vasodilation, leaky endothelial layer, of blood vessels, leading to fluid outpouring in the gut lumen, as well as enhanced peristalsis and mucin production. This is the mechanism of diarrhea, a large amount of third space fluid loss following a food allergic reaction. The process is ultra fast, and the patient, being symptomatic, mostly within half an hour after ingestion of an allergen. The time gap between the exposure and clinical presentation is a useful clinical differentiating point between IgE and non-IgE mediated food allergy. It's interesting to note that the mast cell degranulation does not always involve antigen antibody complex. Physical stimulation like scratching the skin, change of temperature like cold or heat exposure, and sometimes spontaneous degranulation without known cumulatory factors were also observed. Mast cells activation syndrome and nonspecific clinical syndrome associated with spontaneous degranulation and effect of histamine in multiple physiological compartments are being investigated in vivo and in vitro setting. It is also documented in many patients with idiopathic urticaria that emotional distress in itself can also cause mast cell degranulation and urticarial reaction. To continue further, this diagram illustrates mast cells in action in multiple physiological layers, leading to multi-system presentation of an allergic reaction. Some of the mast cells release histamine even in the basal physiological state, and measurement of baseline serum tryptase level gives us some laboratory evidence of mast cell dysfunction leading to degranulation. There are two different phenotypes of mast cells. They are mucosal and connective tissue mast cells. Mucosal mast cells release tryptase, which is packed alongside histamine in the mast cell granule. However, the connective tissue mast cells have additional chymase and carboxypeptidase enzyme that can actively and effectively act against cell wall of the pathogen. These are basically enzymes in its function. It is important to remember that the tryptase level following an anaphylactic reaction should be measured within 4 hours of a severe allergic reaction, but about 18% of the cases, severe reaction may not show raised tryptase level when there is more connective tissue mast cells involvement. In clinical practice, we do see children presenting with multisystem presentation that cannot be explained by single mechanism of action, as illustrated in this slide. There are multiple mediators and cells acting in different compartments which could explain such presentation. One of the important ability of mast cells is its ability to communicate with different defender cells, including dendritic cells, another key player of atopic inflammation. Mast cells, when exposed to continuous high dose of antigen, may start tolerating the antigen without degranulation. This is a proposed mechanism of tolerization process via immunotherapy. The process is facilitated by blocking antibody augmentation in IgG4 level, which compete with IgE and inhibit IgE, thus preventing mast cell and basophil activation and their production of Th2-related cytokines. There is also evidence that after prolonged successful immunotherapy, the mast cell and Treg cell communication, that is T regulatory cell communication, is skewed towards higher number of T regulatory cells and persistent suppression of mast cell activities, which is the underlying mechanism of successful immunotherapy. However, there are questions how long this effect lasts. Dendritic cells is another unique cell which is the antigen presenter that also act as a bridge across innate and adaptive immunity. There are different subsets of dendritic cells classified on the basis of surface marker. To keep it simple, I am not going into the details of dendritic cell subset and its function. The dendritic cells get activated following an allergen exposure. They effectively take the allergen in and present this allergen coupled with MHC class II molecule to naive T cells in the lymph nodes, followed by Th2 skewing, leading to IgE production as opposed to Th1 skewing. This action is facilitated by interleukin 4, another important chemokine that facilitate allergic inflammation. They are found in abundance in the epithelial layer of bowel and can move around freely within the intercellular and subliminal space, catching endless flow of antigen and presenting the antigen to naive T cell. Apart from antigen presentation, they also produce chemokines like interleukin 10, 12, and 23, which attract other effector cells with the help of co-stimulatory cell surface receptor. This immunological crosstalk between different cell surface marker and its relationship with genetic susceptibility to atopic disease is a subject of intensive research. This slide highlights the maturation process of dendritic cells. When activated, they police the subluminal and interstitial space like a guard cell with sole responsibility of antigen recognition. In the great scheme of immune defense system developed over millions of years, these cells are equipped to differentiate between self and foreign protein, where toll-like receptor identification plays the major role. However, the toll-like receptor and its significance in cellular defense mechanism against microbial attack is beyond the scope of this presentation. But I would still like to mention the pattern recognition receptor or PRR and proteins capable of recognizing molecule frequently found in pathogen, the so-called pathogen-associated molecular pattern or PAMP, or molecules released by damaged cells, the damage-associated molecular pattern or DAMP. These are components of innate immunity, but their role could cross over in atopic inflammation of gut, which I will touch upon while discussing food processing and some of its effect in non-Ig mediated allergic inflammation. This is a proposed mechanism where a concurrent infection could aggravate an allergic reaction which is otherwise mild and not severe and quite frequently seen in clinical practice. This slide illustrates how antigen is handled by gut epithelium. As you can see, there are several cells as well as thick mucus coating which are all involved in the immunological process. The simple columnar epithelial cells exhibit physical and biochemical adaptations to maintain barrier integrity, including actin-rich microvillar extensions. Epithelial cell tight junction apically attached and secreted mucins that form a glycocalyx and the production of various antimicrobial peptides. Specialized intestinal epithelial cells known as M cells or microfold cells overlie Pierce patches and lymphoid follicles to facilitate luminal sampling of antigen. M cells exhibit reduced mucin secretion and have modified apical and basolateral surfaces to promote uptake and transport of luminal content to professional antigen-presenting cells that inhibit and inhabit the subepithelial dome in the Pierce patches and lymphoid follicles. Specialized dendritic cells subsets can also extend dendrites between the tight junctions of intestinal epithelial cells to sample luminal content. What is highlighted in this slide is it is a highly effective, and the surveillance is going on all the time to catch the antigen, which is a foreign antigen, either in the food or in the bacterial cell wall. There are other cells which can also present antigen, but dendritic and T cell tango was investigated at length in allergic inflammations. B cells are another key cell in allergic response that produce IgE antibody against food allergen. The antigen IgE antibody together, they can stimulate the mast cell degranulation. There are many subsets of B cell which produce different types of interleukin. Depending on the type of interleukin, it could be either pro-inflammatory or anti-inflammatory. We know about that T regulatory cell or Treg cells are heavily involved in controlling inflammation. Recently, the role of B regulatory cells are explored further. The IL-10 producing regulatory B cells play an important key role in IgG4 induction and IL-10 production, which have significant immunosuppressive function that could help immunotherapy through tolerization. The cluster of differentiation or CD will come again and again, followed by a number that actually define the type and function of different subtypes of white cells and its intended mechanism. For our purpose of understanding allergic pathophysiology, we will focus on CD molecule present mainly on T and B cell. In the following discussion, we will frequently refer to CD4, CD8, CD52, which essentially points towards type of cell involved in the allergic reaction. So far, more than 320 CD clusters are defined and identified in human cells, and it is impossible to remember all these numbers. The synchronized cooperation between T helper cells and B cells is a cornerstone of immunity towards pathogen. But when it comes to allergen, the structure and conformation of allergen finally define and determine the effect of sensitization. I must emphasize that the sensitization is not synonymous with allergic reaction. One can be sensitized to an antigen without being clinically allergic, but all patients with allergy are always sensitized. The class switch of B cells that lead to production of IgE from IgG is influenced by yet poorly understood genetic influence. The mechanism of immune tolerance involves genetic rearrangement in B cells and B cell clone which start producing IgG4, which is basically responsible as a blocking antibody and compete against IgE. When we consider the tolerization process, as happens in milk and egg, when children grow out of their allergy, it is very likely that mutation-associated clonal B cell producing IgG4 overshadow the IgE-producing B cell clone. This could be guided by the antigen structure and conformation and the way antigen-presenting cells interact with T helper cells. As we know, the possibility of outgrowing nut allergy is much less compared to milk and egg. This is again an active area of future research. In this slide, in the center in red, there is CD40 molecule and CD40 ligand, which we will discuss in the following slide while discussing B cell function. T cell in type 1 and type 2 allergic reaction and tolerance. The naive B cell interact with either T helper cell or directly with antigen and transform itself into an activated B cell. Most of the B cells without such stimulation die away. Only 10% cells that survive following T cell or antigen exposure is converted to antibody producing B cells. Now, if you compare the life of B cell with mast cell, you can clearly see the difference. B cells are very mortal. Only 10% of them survives, while mast cells can be immortal and keep on acting as a first line defender. The type of antibody of our interest here is IgE antibody, which is capable of eliminating invaders through mast cell activation. This process involves multiple intra and extracellular communication using receptors on the surface and chemokines within the B cells that finally write down the genetic script through gene rearrangement. Genetic rearrangement within the B cell is the key of IgE production, where B cell producing IgG could switch over to IgE production and follow atopic inflammatory pathway. At this point, the general inflammation or cellular inflammation against antibacterial invasion, antibacterial action changed to atopic inflammatory pathway through IgE production. When there is a defect, either at cellular level or at gene rearrangement level, immune deficiency or excessive production of antibody may occur. This could be secondary to uncontrolled clonal proliferation, as happens in malignancy, or production of excessive ineffective IgE antibody, as in Dobbs syndrome or hyper IgE syndrome. Another example is CD40 ligand deficiency, which stops antibody class switch, resulting in abnormal and inadequate antibody response. Going back to T cell, particularly the CD4 plus and its differentiation is influenced by something called interleukin. CD4 plus T helper cell can differentiate into Th1, Th2, Th9, Th17 and T regulatory cells, and the adaptability is unique. CD4 plus are cytotoxic and can directly kill cells without involving other cells or humoral immunity, which is quite unique. Interleukin is a protein with multiple functions: activation and differentiation of immune cells, as well as proliferation, maturation, migration, and adhesion. So that it means it works at different levels. They are produced by many different cells, and these interleukin act as a messenger so that cells can talk with each other using the interleukin receptor. Where there is inflammation, there is interleukin. Interleukin also have redundant function. For example, one interleukin can do multiple actions. IL-4, IL-5, IL-13, and B cells growth factor can stimulate B cell differentiation. They also have pro-inflammatory and anti-inflammatory properties depending on which type of interleukin we are talking about. Therapeutically, interleukin function could be manipulated, and the receptor blocker can be used to dampen the pro-inflammatory response. Th2, IL-4, IL-5, and IL-13, they are the key players in allergic reaction. The damaged viral epithelium in itself could be a source of IL-4 production. The molecular pattern, which is referred as PAMP and DAMP, which we discussed earlier, are involved in mobilization of inflammatory cells as well as anchoring the cells on epithelial surface, causing further damage. As we are learning more about the epithelial integrity, we are also learning about epithelial microenvironment. This is a theater where multiple actors are playing their role. The symbiosis of resident bacteria, microbiome, substrates that help resident bacteria to survive and grow, prebiotics, their metabolic end product, postbiotics, are all important to maintain a healthy epithelial function. More recent focus is on gut biome as well as fungi. A review article is included in the further reading slide at the end of this presentation. The constant balance of pro and anti-inflammatory molecule is almost like a standoff between two opposing forces. This is further complicated by the fact we are what we eat. That is food fiber, food protein, fermentation end product, etc. that could directly influence integrity of the epithelial barrier, inflammatory response, and how our gut respond to harmless and harmful protein molecule. The interplay between multiple living and non-living factors continue to affect barrier function of bowel epithelium, which is so important in food allergy manifestation. The genetic factors are equally important that decide how our gut immune system is going to respond. Since there are multiple layers of immunological defense, they pretty nicely compensate each other. However, changes in the microenvironment could ignite an inflammatory response that could pretty quickly go out of hand when anti-inflammatory molecule like dendritic cells derived IL-10 fail to contain the inflammation appropriately. Therapeutic strategies to modulate IL-10 is currently being looked in conjunction with oral immunotherapy or blocking antibody or pro-inflammatory interleukin like biologics or receptor blocking antibodies. Gene environment interaction and its effect leading to massive epidemiological shift of allergic diseases across the world over the last 5 decades continued to remain a subject of debate. As a clinician, we must remember the multi-system presentation of allergy. Essentially, this means a structured, robust clinical history so that we could cover all indirect clinical modifier of food allergy like presence of strong family history of atopy, presence of asthma, eczema, and so on. The most important effect of dietary fiber is its ability to modulate T regulatory function and IL-10 production to keep inflammatory process under control. The gut epithelium is always in a state of dynamic inflammation, which is a part of immunological surveillance process. It is akin to a trigger happy sentry in a military outpost. T regulatory cells and IL-10 keep the inflammatory process under control by dampening the unnecessary inflammation that can cause disruption of epithelial integrity. Since in the microenvironment of bowel epithelium, there is constant foreign antigen exposure leading to multiple microinflammation, the short chain fatty acid have the ability to produce IL-18 and enhance epithelial repair. Some controversy as IL-18 could also be pro-inflammatory in selected setting, but I am not going to be going in the details of it as it is not very clear as of yet. These are some of the biologics that could be utilized against different interleukin to block their pro-inflammatory action. Omalizumab and dupilumab is used for eosinophilic esophagitis. However, the role of these blocking antibodies could pose theoretical risk and side effect of breakdown of immunosurveillance, a key component of identification and removal of malignant cells and pathogenic microbe. Next, we talk about antigen. Antigen in food is have the structural and it have a structural diversity and the epitopes. Antigens have a complex structure. In allergy, we are interested in a particular part of antigen called epitopes. These epitopes could be visible on the surface and exposed to immunosurveillance, or it could be hiding deep inside the structure, visible to immunosurveillance only when the 3D structure is altered. Therefore, the factors that alter the structure of antigen can also alter the allergenicity of a particular antigen. Understanding the structure is important to understand how a particular antigen behaves in vivo. The antigen-specific response towards the antigen is determined by the structure as immunosurveillance cells see them. The food processing could change the structure. For example, boiled peanut is less antigenic than roasted peanut. There are several types of food processing that can alter the antigenic behavior of a particular food. Here we will talk about continuous epitopes and conformational epitopes, which is shown in the slide. Another example of heat-induced conformational changes is milk protein. Boiling of milk make it less allergenic, whereas dry roasting the peanut increase or enhance the allergenicity of the protein. However, at the same time, it also enhances the flavor. In clinical practice, particularly in confusing and inconsistent history of allergic reaction to food, it is always helpful to keep food processing in mind and its impact on the allergenicity of food. These are some examples of methods used in food processing industry. When a food protein is joined by a sugar, this process is called glycation that can alter the way the antigen is handled by the immune system. An allergen may become more antigenic and can even directly stimulate a B cell, leading to class switching and production of IgE antibody. The allergen uptake and processing and presentation can also be affected by Maillard reaction. The Maillard reaction sometimes explains when a child with a particular brand experiences a food allergy reaction, while similar product, that is a core antigen, from another vendor or production line who use a different food processing method doesn't show or result in any allergic reaction. Immunotherapy inhibit both early and late responses to allergen exposure. Immunotherapy is accomplished by increasing the allergen-specific IgG, particularly the IgG4 isotype, which blocks not only the IgE-dependent histamine release from basophil, but also IgE-mediated antigen presentation to T cells. We can call this immunoadaptive response. The process of immunoadaptation starts in fetal life when a mother eats allergenic food. The protein allergen appears in the amniotic fluid fairly quickly, and the amniotic fluid is constantly swallowed by developing fetus, and the gut is primed and adapted to these antigens and does not mount an allergenic response when exposed in utero situation while eating food. However, it is possibly far more complex than we understand at present. The sensitization does not always occur through gut, and the other routes like loss of epithelial barrier in eczema and asthma could override the adaptive tolerization and atopic response could be mounted. So, to summarize, for a pediatrician, the clinical history is most important, and linking the history with the pathophysiology of allergy could help us to unravel both usual and unusual allergic clinical presentation. Being a multisystem disease, food allergy does not manifest alone. The role of general pediatrician with gastro and allergy interest, we are in a perfect position to help this complex group of patients. Immunotherapy is future, but the data of its safety, long-term efficacy, cost effectivity, and practicality in general setting is still under scrutiny. This is the slide showing the further reading, and thank you for listening.